My name is Kate Timms, and today I'm going to be talking to you about extracellular vesicles, or EVs, from milk. Despite its common position on our supermarket shelves, milk is a biofluid like and unlike any other. EVs in milk have great potential as a source of biomarkers for assessing animal health in agriculture, for exploring the role of milk in the development and health of the infant gut, and as a potential drug delivery vector. Milk is a complex biofluid containing fat globules, whole cells, casein micelles, and free protein, as well as EVs and other components. This means that several steps are required to prepare milk for EV isolation. First, fat globules and cells can be removed with centrifugation, with whole cells pelleting and fat globules floating, allowing fat globules to be skimmed off. Filtration can further remove fat globules, but removing casein micelles is less straightforward, as their density and size overlap with other components, including EVs. To remove casein micelles, there are several possible methods, including acidification to precipitate micelles, EDTA dissociation, or the addition of chymosin, which cleaves casein and causes it to aggregate. The impact of these techniques on EVs is not yet fully known, but both acidification and EDTA dissociation may damage the EVs. The impact of chymosin is even less understood, but may be the most favourable option for maintaining EV integrity. Even so, as with all techniques, you must carefully consider each variation and compare them to find the ideal workflow for your samples. Finally, free proteins can be removed using size exclusion chromatography-based QEV columns, which come in a variety of sizes to suit your needs. This technique is a quick, reproducible, and pure way of isolating EVs from a variety of biofluids and cell culture media, making it widely applicable across practically any starting material. One of the most interesting avenues of EV research is the impact of human milk EVs on the infant gut. As survival for premature and low birth weight infants increases worldwide, there is a concurrent increase in necrotising enterocolitis. Necrotising enterocolitis is an inflammatory condition of the intestines, which often results in sepsis. It is fatal in around a quarter of affected neonates, making the potentially protective impact of breast milk EVs an important area of research, especially as these neonates often do not have access to full-time breastfeeding or even any breastfeeding at all. In mouse models of necrotising enterocolitis, milk EVs were found to be protective against mortality and reduce disease severity. This could be through the ability of milk EVs to increase intestinal epithelial cell proliferation, improve goblet cell function, and improve the tightness of the intestinal barrier. These properties may also be helpful in other inflammatory intestinal disorders, such as colitis. The positive impact of milk EVs given orally in mouse models suggests that they may reach the intestine intact. This could be because the infant digestive system is gentler than that of adults, but artificial cargo from milk EVs given to adult mice was detectable in distal organs, suggesting survival in adults too. This makes milk EVs an attractive drug vector, an idea which is currently showing promising results in early in vivo studies. Despite their great potential, studies on milk EVs are lagging behind those on other biofluids. At the time of recording, only 4% of biofluid EV studies on Web of Science were related to milk EVs. This makes milk an exciting biofluid to work with, as much is yet to be discovered, and several key questions remain. The first is whether milk EVs can be purified to a high standard without damaging the EVs. As discussed earlier, the composition of milk makes purification a challenge, yet purity is essential for downstream uses, particularly drug delivery. Then there is the question of whether milk can be stored in such a way that it maintains EV integrity and functionality for therapeutic purposes. Breast milk is often stored in freezers for use by premature and low birth weight infants. But does this thawed and warmed milk adequately reflect the original breast milk before storage? Another important consideration is whether milk EVs are safe for long-term treatment. Milk EVs have been shown to be non-toxic in short-term studies in vivo, but no long-term studies on toxicity have been conducted. The potential exposure to low-level toxins could accumulate over time, either from endogenous EV components or from the environment and the donor animal or person. Even if they are non-toxic, how well would cross-species milk EVs be tolerated in neonates and in adults? Non-human milk is less optimal for neonatal health than human breast milk, and many adults do not tolerate cow milk. It is not yet known whether cross-species milk EVs would provoke the same reactions. Finally, will the promising results from drug-loaded milk EVs translate to successful clinical trials? The first clinical trials on milk EVs have yet to begin, so it will be interesting to see how these therapeutics perform as they jump from bench to bedside. Whatever the outcomes of these questions, milk EVs are an exciting area for research and therapeutics.